oplaufen.
I open this academic ceremony in which Mrs. Lydie Barbeau will defend the academic thesis Harnessing Autophagy in Cancer, Leveraging Biomarkers to Improve Personal Therapy and Treatment Efficacy. I welcome you, of course, and wish you lots of success together with your paronyms. I also welcome Professor Ruschop and Dr. Curtis, your promotion team, and of course the members of the opposition, whom you will meet later, and family and friends here present and online. But before we start the opposition, I give you the opportunity to tell family and friends what your thesis is about.
Thank you, dear Prorector. Dear Prorector, Corona members, friends, family, and colleagues, thank you for being here today. And during the coming 15 minutes, I will present an overview of my thesis entitled Harnessing Autophagy in Cancer, or in Dutch, Het benutten van autofagie bij kanker. So first of all, what is cancer? So the human body is made of small organs, themselves made of small units called cells. And normal and healthy cells participate in the good functioning of the human body. Due to factors, these normal and healthy cells can transform into disease cells, here a tumor cell. The accumulation and growth of these tumor cells form a mass called a tumor, and the disease named after cancer. Tumor needs a lot of oxygen and food to grow and progress. Food and oxygen are provided by blood vessels around or within the tumor. Due to the fast growth of tumor, food and oxygen are limited. A gradient of oxygen is created within the tumor. So here you can display it by these blue cells and those red cells. So the closest you are from the blood vessels, the more oxygen you have. So here in red. This is called normoxia. The further you are from the blood vessels, the less oxygen tumor cells have. So here in blue, called hypoxia. This lack of oxygen, or hypoxia, is a signal for a signal and a characteristic of cancer. Lack of oxygen or hypoxia is associated with therapy resistant for patients, so medication not working for the patient. It's also associated with a higher chance of having tumor cells spreading elsewhere in the body. And overall, patients have a worse prognosis due to this characteristic. Lack of oxygen or hypoxia is stressful for cancer cells. To survive in those stressful environments, cells turn on a survival mechanism called autophagy. Autophagy means self-eat in Greek. So the cells are literally eating themselves to survive. So autophagy is a recycling mechanism, which is essential for the cells to recycle garbage and reuse this product of the recycling to make new substances to survive. For cancer cells, autophagy is really essential because it provides new nutrients to grow and spread elsewhere in the body. So, luckily, there is a drug to block autophagy and to limit cancer progression. This drug is called chloroquine. Chloroquine is used nowadays to treat malaria and rheumatoid arthritis disease. So it's a commonly available drug on the market. The consequence for the tumor when chloroquine is given is an accumulation of garbage within the cells. And this leads to their death. However, not all tumor cells respond to this treatment, leaving behind tumor cells behind, so here displayed in blue. So how us or in the clinic can predict whether a tumor and a patient is going to be resistant or sensitive to the treatment? So this is what we have addressed in this thesis. In the chapter 2, we have identified biomarkers and biomarkers or biological signals and tumor that are sensitive to chloroquine. And we validated those biomarkers and those tumors in the lab to show how they can predict sensitivity to the drug chloroquine. We also found that autophagy was naturally absent in some cancer and showed that we can use this absence of autophagy as a biomarker to predict prognosis for patients. As last, we described it an autophagy-related protein which can identify a subtype of tumor which does not have immune cells within the tumor mass, and highlighted the value of this characteristic for future therapeutic application. First, let's focus on the first chapters and the biomarkers and tumors that indicate sensitivity towards chloroquine. So, first thing we did, we screened the literature and identified different tumor types, here in different colors, and signals that could predict whether a tumor is resistant or sensitive to the drug chloroquine. Then we concentrated our work on brain tumor to confirm our findings. To study cancer, we used cancer cells that were extracted from cancer patients, and we cultured these cells in the lab. With these cells, we can predict whether a tumor cells, and eventually a patient, can be resistant or sensitive to any drugs. So what we first confirmed is that indeed the tumor cells can grow in a dish, in a petri dish in the lab, and they do. Then, for the purpose of our work, we used chloroquine as a treatment, and we treated our cells, glioblastoma cells, with chloroquine to test if they were sensitive to this drug or if they were resistant. After treatment, we observed two profiles of cells. On the left graph, one profile where the cancer cells did not die with the treatment, so here in pink. They grow as fast as the cells did not have a treatment, so the resistant cells. And on the right graph, what we can observe is that the cancer cells which have been treated with chloroquine died. So also a reduction in the percentage of cells that were alive, so the sensitive cells. So if we extrapolate this data from the lab to patient, this data suggests that in the future, if in the clinic we can screen the patient and test the patient for the sensitivity of this drug, patients which will have a similar profile as the sensitive cells can be given the drug chloroquine because we know they will be sensitive to the treatment, and eventually we can improve their outcome. On the other hand, resistant patients will not need to get the treatment because we know they will be resistant. So the question is now, how can we make this resistant patient sensitive to chloroquine? So tumors from those resistant patients are not dependent on autophagy to survive, despite being in a challenging environment. So we thought to force the cells, the cancer cells, to be dependent on autophagy. To do this, we use drugs that are already available on the market, so that those drugs are used to treat other diseases. So if our hypothesis is correct, if we give this drug X to some resistant cells, they will turn on this autophagy pathway, be dependent on them, so that we can add the chloroquine treatment, and the cells will eventually die. So that's the hypothesis. So now we tested this hypothesis in the lab. So first, we confirmed that without any treatment, cancer cells died. Then we gave to the cancer cells either the drug X or the drug chloroquine alone. only 50% of the cells died. So which is, the drug has already an effect on their own, but not sufficient. So then we combine both drugs. And what we observed is a drastic increase of cell deaths. A lot of cancer cells died because of the combination of the drug. That means that we highlight here a potential new therapeutic approach to treat patients by using a combination of drugs that are already available on the market that can, with which we can treat resistant patients and eventually improve their outcome. So far, I have presented you cancer cells which have, or tumors which have autophagy in them. But what about cancer cells which does not have autophagy? So we collected clinical data from patients, and we separated them in two groups. On the graph, you can see a group where you have patients with a tumor with autophagy, and then also a group of patients with tumors without autophagy. And what we observed in those two groups of patients is that in blue, the patient with a tumor with autophagy had less chance to survive cancer, whereas the patient who had a tumor without autophagy, so in pink, had a better chance to survive. So how is it possible that just having or not having autophagy make such difference for the survival of the patient? So we studied the tumors from those patients, and we observed that this difference of survival came from the composition of the tumor. So for the patient in blue who had tumor with autophagy, the tumor mass was full of cells that were resistant to treatment. Whereas the group of patients which had a tumor without autophagy, in pink, had tumor mass only made of sensitive cells. So what we show here is that autophagy is so essential for the cells that, so in blue, that if you remove autophagy from them, those one died. And what's left behind is only a tumor mass with sensitive cells. So that illustrates the fact that not having autophagy can be a biomarker to identify patients responsive to treatment so that we can improve and develop personalized treatment for those patients. So the conclusion from this thesis, and I hope I convince you that autophagy is essential for cancer cells to survive. We have identified tumors and biomarkers that predict sensitivity to chloroquine treatment. We also show that autophagy dependence can be changed and increased by using drugs that are actually available on the market. We also show that autophagy can predict patient prognosis with having or not autophagy. And at last, something that I've not showed here, but which is present in the thesis, is that an autophagy-related protein can be a valuable biomarker to distinguish tumors which have or not have immune cells, which can be, for the future, an indication to give immunotherapy or not for patients. So overall, we highlighted that not having autophagy or inhibiting autophagy increases the therapeutic efficacy in preclinical studies and in cancer patients. And we reinforce the importance of biomarkers-driven strategies to optimize personalized treatment for patients and improve their outcome. So with this, I end my presentation, and I give the word back to the prorector.
Thank you, dear candidate, for a nice presentation. Then we start the opposition, and we do that with the chair of the assessment committee, Professor De Ruijser, whose expertise is radiotherapy and oncology in the Maastricht Clinic of the University of Maastricht. Professor De Ruijser.
Thank you, Mr. Pro-Rector. First of all, I want to congratulate really great work. And I know that the thesis is not only work in the lab, but also a social adventure. So congratulations for that. But I'm here to ask you some questions, of course. And I want to discuss with you chapter 6. And as you may know, I work the whole day with lung cancer patients. And I wanted to know what exactly is the basis that low MAP1 LC3 is associated with low HLA class 2 expression and also low immune infiltrations. Do you have an idea about the mechanisms between both observations?
So, highly esteemed opponent, thank you for your kind words and thank you for your questions. So we have research indeed, so that the observation that low LCCC was associated in low HLA was done in clinical data from patients. We have tried to understand the mechanism behind the fact that you have low infiltration also on immune cells. What we can think is that, LC3C might not be a protein. It's a protein associated with the autophagy pathway, but not directly used in the main mechanism. But we can imagine that, LC3C has a role in degrading eventually HLA protein, which has already been shown that autophagy is involved in the degradation of HLA molecules, but also in peptide processing to be presented on the surface of the tumor cells with HLA. So we can imagine that LC3C might be involved in this, in this degradation pathway. Besides, LC3C is also, associated with xenophagy. So like presentation or like in, in the degradation of microbes or anything. So it could be that it's also mechanisms of defense for the tumor cells, also like for being attacked from foreign bodies or anything like that. Mm-hmm. So, and the fact that you, the fact that there is less infiltration might come from the fact that actually not having HLA at the surface of the tumor cells does not trigger necessarily, or does not trigger enough an immune reaction, at least from the HLA class.
Mm-hmm. And do you think there's a causal relation between the expression of the, call it biomarker, so the MAP1, and having or not having not only immune infiltration, but also that the T cell will recognize the cancer cells as being foreign?
Both can be, like, we have not really defined who is the chicken from the eggs. But what we can imagine is like first an event where cancer cells could use LC3C or low or decrease by themselves LC3C to not have HLA expression to decrease the immune reaction. But it could be also the fact that if you do have an LC3C expression, you will have an immune infiltration. So the cells that are actually within the tumor, cells having LC3C might be killed by immune cells, and the tumors will be replenishing the the emptiness by cells not having LC3C. So it could be, what's the egg from what's the chicken from the eggs? It's always the question.
And if you go back to chapter 3 and you have identified some drugs which may influence the whole complex chain of autophagy, with which drug would you combine immune treatment in patients with a low MAP1 LC3 expression?
So in chapter 6 we have used the drug vorinostat if I'm not mistaken, we have used the drug vorinostat to show that we can increase actually HLA class 2 expression. So,
but that's, I will rephrase my question.
Yes.
If you want to go for a clinical trial, then it's not only about more HLA-2, but it's also more immune infiltration. And also, you want to combine this with current treatments most of the time, I presume. And if we have a cold tumor, so without HLA expression, without immune infiltrations, we typically treat those patients with dual checkpoint inhibition, like an anti-CTLA-4 together with an anti-PD-1 or PD-L1 antibody. Which drugs would you think, and a short answer please, would be preferential and why? But really short answer.
I mean, if you wish to have an immune reaction, I would give an anti-PD-L1, so which will trigger, how do you say?
Would you use chloroquine?
In that sense, I'm not sure. Yeah, blocking autophagy could end. Yeah, I'm not sure because we tried here. If I'm not mistaken, chloroquine did not really change the expression of HLA actually. So we could try, but from my knowledge and what we have done here, I'm not sure chloroquine will enhance the immunotherapy in that case. But it is worth to try. But in that case, yeah, I would not be 100% confident that will work, actually.
Thank you very much for your answer, and I give the word back to Mr. Pro-Rector.
Thank you, Professor de Ruyse, for your position. And we come to the following opponent, who's also a member of the assessment committee, that's Professor Koppes. And Professor Koppes' chair is Radiotherapy and Radiation Biology of Normal Tissue in the University Medical Center in Groningen. And he's online, and thank you for coming all the way from Groningen.
Okay, thank you. Dear candidate, first I want to compliment you and your whole team actually with the very nice book. I learned a lot about autophagy and all the related issues in cancer. And actually there's a huge amount of data in there. I think it's really impressive, all the work that you did in the time that you had. But I still, of course, have also some questions. So I would like to go with you to chapter 3, and especially figure 2 in chapter 3. And in this chapter, you want to see if EGFR and P63 are able to predict whether or not chloroquine would make sense to treat a patient with chloroquine, right? So then you use a whole bunch of cell lines where you look at the EGFR expression and then separate them in sensitive and resistant tumors. And and then you decide on whether they're sensitive or resistant, whether or not they should be treated with chloroquine, right? So if you look at figure 3A, you can actually, if you look at that figure only, would you still think that EGFR would be a nice biomarker to separate the two populations?
Highly esteemed opponent, thank you for your kind words and thank you for your question. Indeed, based on figure 2A of chapter 3, taking alone EGFR expression will not be an ideal biomarker to distinguish whether tumor cells will be resistant or sensitive to chloroquine, as we can observe 3 cells which are actually resistant to chloroquine have high expression of EGFR, as much as its sensitive cells. So on alone, EGFR expression, to me, would not be a good biomarker to predict chloroquine. That's why we try to find another, another biomarkers, another way to predict the sensitivity.
But in the study, you still described that you did a clinical trial with patients based on the EGFR expression, right? And then you indeed find actually more the opposite, that EGFR would respond better, the tumor would respond better to chloroquine even if the EGFR receptor is not there, right? And so that I find a bit weird. Would it not be better to look at P62? And why did you not look at P62? Because the graph figure 3 really shows you very nicely that these are very nicely separated, right? The resistant and sensitive population.
Yeah, so we stick to EGFR, first of all, because EGFR expression is one of the biomarker which is actually currently used for patients to determine the subtype of the glioblastoma for patients. So it's already something in clinic. And second, using P62 on its own, to my knowledge, has never been shown to actually determine whether a tumor is resistant or sensitive to chloroquine. And P62 is not a biomarker currently used in clinic to determine whether a patient can be, responsive or sensitive to chloroquine. So it is something that could be implemented also in the clinical daily routine of, daily routine for the, in the hospital. But that's why we stick with what we knew. EGFR is already a biomarker for, for patients, and knowing that EGFR V3 patients also are, from previous study, are actually sensitive to chloroquine. So using EGFR is still a valuable marker if it's mutated or not, and without any mutation, P62 might be a good indication. But
it's a bit different in your. But okay, but would it be possible still? You may have the material still of these patients, right? To measure the P62 and still see if you can correlate that to the chloroquine response, because it looks very promising. Right?
Yes, indeed. Like we do have, tumor patient from the cohort used in one of our previous study, but also shown in figure 4. And the plan was to stain actually those patient tumor slides for P62 to correlate our findings from figure 3 and to extrapolate these findings to patients also. But this is a work which is actually ongoing.
Okay, that's great. I would love results in the future, but I have another question. This is about chapter 4, where you very nicely describe 2 different types of drugs that could be used in combination with autophagy inhibitors. But you do this nice CRISPR screen on cell populations, and then you come up with two pathways, right? The abbreviation I can see, the FIN and the PRKCH, right? Maybe I pronounced it wrong, but okay. So my question exactly, because I see a big cloud in your CRISPR screen under hypoxia, right? And how did you come to pick these 2 markers? Because there are so many other dots that might be interesting as well, right?
Yes, indeed. There are a lot of points in a big cloud, and how. So the way we determine those 2 hits. So like in the supplementary figure table S1, this is the list of genes that actually came up after statistical analysis. So those genes are the ones coming up from being the top 20 genes being changed. And we looked for drugs that could actually target those individual genes. So it's the drug-tested column in the table S1. And we tested individually all of those drugs to see if whether or not we confirm the data from the screen by using those drugs and see if the cells were sensitive or resistant to this drug. So, and PRKCH and FIN were actually the genes that where we used dasatinib and tamoxifen. So So like FIN and PRKCH are isolated from the screen, and we saw effect on dasatinib with dasatinib and tamoxifen. So we speculated that FIN and PRKCH from the statistical point of view were the target that were actually identified, but dasatinib and tamoxifen have side effect and target other genes. So it could be still that it's a coincidence that we found those 2 genes. It could be also other genes, but from the statistical point of view, those are the 2 genes that were responding probably to those that were indicating a response for dasatinib and tamoxifen.
Okay. So partly science and partly gambling, right? But okay, very nice. I think my time is over. I'd love to discuss a lot more, and you'll give the word back to the chair.
Thank you very much, Professor Koppes, for your opposition. Now we come to the following opponent, who is also a member of the assessment committee. That's Professor Langen. He is a professor in skeletal muscle biology and lung pathology at the Department of Respiratory Medicine at the University of Maastricht. And today he has also accepted the role of secretary of this committee, for which I'm very grateful. The floor is yours.
Thank you, Mr. Prorector. Dear candidate, first of all, also my congratulations with the completion of this very elegant work described in your thesis. And of course, I would like to extend those congratulations to your promoter and co-promoter. Aussi, mes félicitations à votre famille avec la soutenance de votre thèse. And I would like to finish by stating my compliments for the clear and focused way in which you have described all of your findings and brought them together in the discussion of your thesis, highlighting the importance of your work in the context of the progress in the field. After all these compliments, of course, I should definitely use the rest of my time to ask you some questions. And I will start with Chapter 4. In Chapter 4, you use a very elegant, unbiased strategy. It was already referred to by Professor Koppes to apply this kinome screen. And there, a number of druggable kinases are identified. And you take two of those that can be targeted with tamoxifen and dasatinib. And my first question relates to the in vitro model in which you then start to investigate the impact of these two inhibitors. And the main theme of the paper is, of course, that you are investigating autophagy dependence, especially of hypoxic cells. And I would like to know to what extent this model system was really suitable to address that. And let me specify what I mean with that question. If you look at figure 2a, there are experiments done with the U87 cell line, and there you basically already compare the impact of chloroquine on cell numbers under normoxia and hypoxic conditions. And those are the light and gray, the light gray and the dark gray lines. And to my surprise, for this cell line, the impact of chloroquine actually seems to be much bigger in normoxic than in hypoxic conditions, whereas the theme of the approach and the underlying hypothesis was that it would actually be the hypoxic cells that would be addicted to autophagy. So can you explain a little bit more what I'm looking at there and to what extent that model was still suitable to pursue the overall hypothesis?
So, highly esteemed opponent, thank you for your kind words, also in French towards my family and to me, and thank you for your questions. Yes, indeed. So we use the U87 model, which is a commonly used glioblastoma model in the lab, easy to culture. But I agree with you indeed. Like under, under normoxia, chloroquine has a strong effect compared to hypoxia, even though the X and Y, the Y-axis are different in normoxia and the hypoxia also. So that's also where the difference may lie. Also, it's like also the Y-axis.
Yeah, maybe I can follow up on that. I noticed indeed the difference in the Y-axis, but then still, if you express it as a percentage change, the percentage is bigger in the normoxic compared to the hypoxic conditions. But in both conditions, you do see this effect, right, of chloroquine. I suppose that also would make sense from the point of view on how cells do respond in glioblastoma patients to chloroquine. So I can imagine that would be that was sufficient for you to continue with subsequent experiments. And then in figure 4, you look at the autophagic flux, and I already know that some of the other opponents may ask you on how that experiment is performed, so I won't go into that, but I would like to know the outcome of that experiment, because there's a couple of comparisons that you make, and one of the comparisons related to the autophagic flux is the extent to which this is induced or elevated by hypoxia. And there you see a very nice increase in autophagic flux by hypoxia. But then to my surprise, when the kinase inhibitors are added, specifically tamoxifen, this increase is gone and is actually not further increased, which would have been my expectation. Can you explain a bit more those results and how they relate to autophagy addiction? Indeed, like if we expose the cells under hypoxia, so in figure 4a, indeed, like what
as expected, hypoxia induces an autophagy, an increase of the autophagic flux for the cells to survive. If we had tamoxifen in pneumoxia, this one is increased. So we could also somehow show that under pneumoxia, the cells are inducing autophagy actually to survive when we stress them with tamoxifen. And under hypoxia, this one is actually decreased. But decreasing autophagy is also something that, how do you say, you could expect also a slowdown of the flux that could induce actually cell death. So like lowering the flux of autophagy could also accumulate, lead to an accumulation of garbage eventually on the cells and induce cell death. So actually increasing autophagy, if you would have, they would be dependent, especially with the tamoxifen.
And then subsequently additionally sensitive to autophagy inhibition. That's actually what I would expect it to happen there.
Yeah, exactly, yeah. But how do you say, it's not necessarily because you have a high flux of autophagy that you are depending on it. Like we have showed in previous paper that actually having low flux of autophagy, it's what makes the cells dependent on autophagy, because if you inhibit it, the cells die. So having high or low autophagy does not necessarily mean that you are actually sensitive to the drug, or more or less sensitive to the drug.
Okay.
But it's just you have the bare minimum to survive, actually. So that could also be an indication. Either the cells are so dependent that they rely on the bare minimum, or actually here, tamoxifen is lowering the flux to actually induce an accumulation of garbage within the cells that will induce cell death, actually.
Very well. I'm satisfied with that answer, and I also understand that that was sufficient to continue to in vivo experiments. Hopefully there will be some questions about that. But for now, thank you for your reply, and I would kindly return the word to the prorector.
Thank you, Professor Langen, for your position. We now come to the following opponent, is Professor van Engeland. She is professor in pathobiology of cancer at the Institute GROW in the University of Maastricht.
Thank you, Mr. Prorector. First of all, congratulations to you and your supervisory team with this beautiful thesis. I've learned a lot about the biology of autophagy and surrogate markers that you try to find to indicate autophagy. But I would like to start discussing the concept of biomarkers with you. You've used that word a lot in your title, in the thesis. And I would like to ask one of your paranymph to read proposition 7. Seven.
On the mic.
Identifying and implementing reliable biomarkers to predict treatment response and improve patient outcomes are at need.
Okay, and the reason why I want to discuss biomarkers is that you might be aware only less than 1% of all biomarkers that are being described in scientific literature are being used in the clinic. So translating biomarkers to the clinic is neither swift nor easy, and that's a tremendous understatement. So what is a reliable biomarker in your opinion?
Highly esteemed opponent, thank you for your kind words and thank you for your questions. So for me, the ideal biomarker would be something that, a reliable biomarker would be something that actually predicts whether the prognosis or predict the prognosis for the patient or a prediction for a treatment response. So it should be something that is actually easy in clinic also to assess and something that actually does not give a lot of doubt or like not in the gray area of the range whether it's going to the drug is going to work or not, or is the patient is going too long to survive, short or long, but something which gave much more a black and white answer also, especially for the patient to give them the good news or the bad news, actually, also in terms of prediction for treatment response or their prognosis also.
Okay, and then in chapter 2, you did a review on biomarkers for autophagy. I didn't find the information on how you did the selection of the papers that you used. But then in the discussion, you mentioned that it was a systematic review. So how did you perform the selection of papers? And I think that's important to actually value the list of biomarkers that you presented, right?
Yeah. So the way we performed the selection of the biomarkers were based on in vitro or let's say and clinical data, based on whether the giving the the biomarkers, whether we saw a difference in cell death or not, whether the biomarkers, when treated with chloroquine, induce a change in autophagy flux, but also a difference in terms of outcome, whether cells will die more or die less, actually, from the treatment combined with chloroquine. So whether knocking out or inhibiting the gene with a drug combined with chloroquine will induce enhance cell death compared to treatment alone, actually.
Okay. Did you consider the level of evidence? So whether this study was validated by independent research groups, or did you consider the power of the studies and the number of samples or cell lines that have been used?
For some biomarkers, that was actually possible. So like for the BRAF mutation, that was already several times, published and documented. But for some others, that was only a matter of one publication. So it's something that should be addressed further also by other, by other scientists. But for most of those papers, they were not interested in whether the cells were depending on autophagy or not. They were just looking at whether these genes had an effect on other pathway and at chloroquine during their experiments. So that was not necessarily the aim of their, the main aim of their research, actually. So it's us by looking at, focusing, on whether the drug or the, combining with chloroquine, whether there was an induction increase or not of cell death, actually.
Okay,
so it was.
Thank you. So we discussed the reliability of biomarkers now. Can I ask the other paranym to read out proposition 8?
Repurposing drugs and implementing biomarkers-driven stratification of patients represent promising strategies to rapidly improve therapeutic efficiency at a lower cost.
Thank you. So now I want to discuss the word implementing. Can you very shortly elaborate on what you think is necessary to implement these biomarkers?
So, like you mentioned, some of them has been, briefly shown in the literature. So I would focus on those biomarkers. I would, study a bit further. For the one that have been proven to work, try also in vivo, because for most of these biomarkers, in vivo experiment has not been done. So I would further also study those biomarkers in vivo to see, to see if they could be implemented or not towards patients, and also use clinical data actually that are available to see if those biomarkers actually, with the data that we have, indicate a better outcome or not. I don't know if.
Okay, thank you. Thank you for your answers. I'll return the word to the pro-rector.
Thank you, Professor van Engeland, for your opposition. We now come to the following opponent, that's Dr. Moute. His expertise is autophagy in the University Medical Center of Groningen. And thank you for coming all the way from Groningen.
Yeah, thank you. Dear candidate L, I also first want to congratulate you to this very nice thesis, and also extend my congratulations to the co-supervisor and supervisor. I would like also to get your paronyms a bit to work for the proposition number 6, please.
Autophagy is an underestimated molecular pathway that could provide valuable opportunities for targeted cancer therapy.
Yeah, so I'm also from the autophagy field, so I'm all pro-autophagy, but then I also agree with you in chapter 2. In the conclusion, you say that for now it's impossible to measure actually autophagy in especially in tumor samples. So how do you see how should we use autophagy? Are we there yet to use autophagy in therapy? Should we even pursue it?
Dear esteemed opponent, thank you for your kind words and thank you for your questions. Yes, I still stood behind my words. We still should use autophagy as a biomarker, even though we are still not capable of measuring the flux or like the flux of autophagy in patients. Actually, so one of the possibility to use it is like to take different biopsy from patient, which is actually not impossible in terms of ethic for patient. But I guess, like for example, using P62, which is an indirect way of measuring autophagy, for instance, could be used as a biomarker for patient for like to measuring the autophagy flux. So we could use that. But other current alternatives are not there. So like the current possibilities are used using colors or using genetic modify system like the mCherry GFP system that you cannot inject in patient actually. So yeah, there are a lot of work to be done in that part, like to determine the autophagy in patient actually.
Okay, and then because you also mentioned that taking biopsies is also not really something that the patient really wants. So do you think there is a way that we can maybe measuring whether the autophagy sort of treatment works in blood or something in fluid that is close to the tumors, that we can actually use then this as a readout whether or not we modify autophagy at all?
That's something. Using liquid biopsy is something that is more suitable for the patient, but the thing is, like you could measure, indeed, you could use liquid biopsy to eventually measure autophagy, but that will be from the blood cells of the immune cells. So which is not necessarily something that you're interested in. You will be more interested in the tumor themselves to know what's the autophagy status from the tumor cells. But you do have, tumor cells still in the bloodstream eventually sometimes. So you could eventually try to, if they are existing, or like if you can isolate enough tumor from the blood vessels to check if autophagy is inhibited or not.
Okay, that's fine. Okay, so then I want, because basically almost in your whole thesis you use chloroquine or chloroquine, which is a very late autophagy inhibitor. Actually, it's a lysosomal inhibitor. So do you then think that everything, all the basic features you see is then due to the cells that needs the recycled material, or rather the removal of damaged material, which is then the more important aspect of autophagy that you block in your tumor systems?
So using the properties of chloroquine blocking the lysosome, or?
Yeah, what I mean, because the autophagy pathway is on the one hand you remove damaged material, right? And then you also recycle material. And since chloroquine is a very late inhibitor, I'm wondering, would you then agree that you need actually the recycled material for the autophagy, for the tumor cells to survive, or actually the feature that the removal of damaged material?
I guess I think, to my opinion, would be much more the recycling mechanism, because otherwise you will have an accumulation of garbage, and the cells will stress and suffer from this accumulation. So I think, it's much more the accumulation of garbage, I guess, that the cells will suffer from. But chloroquine has also a side effect, also that does not only affect lysosomes. So it could be also like what the effect that we see now, or the fact that we use chloroquine, is not directly linked with blocking autophagy, but could be also other side effects that we have not studied here also. So it could be also an acidification or something like that from the lysosome that stress the cells also. So
okay, and then at least in your cell system, which is easy to manipulate, have you ever tried any other autophagy inhibitors, sort of early autophagy inhibitors for the PI kinase, or?
no, we have not tried, simply because chloroquine is a drug which is actually possible to be given to patients. So we stick to drugs that were really possibly given to patients in the future. We did not try to inhibit other part of autophagy. We use, for some experiment, shRNA against LC3B as a, I would say, proof of concept for real autophagy inhibition, but we stick to drugs that could be translated for patients, actually. We did not try other drugs. But that's something that could be also interesting to see if it's something related to early autophagy, or is it a side effect of the chloroquine, actually, or like odd side effect, other, I would say, mechanism that chloroquine has.
Okay, thank you very much, and I give the word back to the corrector.
Thank you, Dr. Mouten, for your opposition. And we come to the following opponent. It's Dr. Dubois, and his expertise is precision medicine at the Institute GROW at the University of Maastricht. Dr. Dubois.
Thank you, Mr. Prorector. So, dear candidate, also my congratulations for your thesis, and I want to extend this congratulations to your promotion team, your family and friends. I also have some questions, obviously. So in follow-up of the previous opponent, where you indicated, or where it was indicated it's difficult to measure autophagy, we don't know exactly how it works. You have shown in chapter 5 that loss of ATG12 is actually leading to better outcome in head and neck squamous cell carcinoma. And in your presentation, you also showed suddenly like a hole in a tumor, well, as kind of a cartoon. And you have shown in this chapter that you actually have a reduction in hypoxic fraction. So I have the solution for all these problems here. You don't have to measure autophagy, but measure hypoxia. Would that be a way out?
Dear esteemed opponent, thank you for your kind words and thank you for your question. Yeah, I know your work in the field of hypoxia and that you have HAP available, ready to use in the lab, and that are actually transferred in patients. But yeah, actually, yeah, hypoxia is something that is actually giving side effect or like giving resistance to the drug. So, measuring the amount of hypoxia is something that could be also used, but I would still combine it with, how would you say, with autophagy, simply because chloroquine or like at least autophagy inhibitors are available and accepted to be used for patients, while I know that HAP, barely any HAP are used, so hypoxic activatable broad drug are ongoing towards patients. So to be used in patient, actually. Yeah. So
well, I expected that answer, and obviously it's needed to also look at autophagy. Otherwise, why have you been working for years on autophagy? Yeah. But interestingly, in chapter 5, you only looked at HPV negative head and neck squamous cell carcinomas. Do you think there's a role for ATG12 or loss of ATG12 in HPV positive tumors, which do outcome-wise already better?
So HPV positive tumors are completely different, or etiologically they are completely different than HPV negative. So they are triggered by a virus, which is genetically different than having HPV negative. So whether ATG12 has a role or plays a role in the tumorigenesis of those HPV positive has to be proven, but it could be the case, or it could be something completely different. That would have to be tested, actually.
Yeah, okay, because that brings me neatly to chapter 6, where you showed that reduced LC3C does play a role with viral in one of your correlations that you have done. Well, I'm not going to ask a question on viruses, because I don't know anything about those. But you showed that reduced LC3C leads to a reduction in HLA class 2, and then you make a leap that this actually may result in dampened CD4 T-cell immune recognition and survival advantages in one of your correlation matrices. What experiment could you set up to actually prove this? Because you did not provide any proof of this so far.
Yeah, so the ideal experiment would be to use T cells, actually, like to co-culture tumor cells with or without SC3C and do a co-culture with T cells. But the problem is that, I would say, we tried to do co-culture with other immune cells, which did not come up with good success, but that was actually easier than using T cells, actually, because T cells, if you can have an immune reaction, how would I say, you need a match between the tumors and the T cells, so it has to come from the same patient. And having T cells, like for this study, we could not have a tumor from the patient and the T cells from the same patient to prove our concept. So the ideal word to prove our word would be, prove our idea would be to have a tumor from a patient and being able to have T cells from this patient to see if you do have an immune reaction due to loss or I would say increase of SC3C and not because of mismatch between the T cells and the tumor cells, actually.
Right, but the human tumor cell, or the tumor cells, is based on the fact that you used a human SW900 lung cancer cell. Why not using an LLC murine cell, where you actually have also easy T cells, and try to test that in an immune competent in vivo setting, for example?
Yeah. So we screened several tumor cells actually in the lab, available in the lab, and unfortunately only these cells are SC3C expression. So SC3C is not, how would I say, spread, or like it's not present in a lot of tumor cells. So, and those cells, I'm not sure we have tested them, or if we did tested them for SC3C expression.
Well, suppose they have expression. What experiment would you set up then?
Yeah, exactly. So if so, then like do a co-culture with T cells to see if suppressing or increasing SC3C, do a co-culture in T cells in vitro, and then if it's possible indeed in immunoproficient mice, inject those tumor cells and see if the level of immune reaction is increased or decreased because of those tumors, actually.
Thank you, Dr. Dubois, for your opposition. Dear candidate, Ms. Lydie Barbeau, the time appointed for the defense of your thesis has passed. The degree committee will now withdraw to discuss the quality of your thesis and the way you defended it, and I ask you and your company to await the results of our deliberation and our return in this room. I suspend the meeting.
The PhD defense has now ended. The degree committee will debate the candidate's performance behind closed doors. This process usually takes about 10 minutes.
Long road to the south side. 7 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the line, I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. Cause I'll go the extra mile. Long road to the south side, 10 miles in my rearview mirror, I know what it felt like. My goals only getting clearer, east side to the west side. No place like home, if it's questions that you've got, go the extra mile and that. On the dash, got my laces tied. Long road, I don't waste no time. Break rules because fate decides. With the team and we chase the light. I make a move, fall down, shake it off. I hate to lose, bad branch, break it off. No room for negativity, praise and love. Prepare for deep part cause we're taking off. Get the mileage, traveling so far from home. Gathering knowledge, other connections. I put the time in to get Even though these nights seem long, I know that I am never on my own. Know you won the battle once you've grown. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and not. Long road to the south side, 10 miles in my rearview mirror. I know what it felt like. My goal's only getting clearer. East side to the west side, no place like home. If it's questions that you've got, go the extra mile and not. What's on the line? I know you know that I can't read your mind. Years can't fly by now, don't waste all your time. 'Cause I'll go the extra mile.
I reopen this academic ceremony. Ms. Lydie Barbeau, the degree committee here present has assessed the quality of your thesis and the way that you defended it. And in view of its positive verdict and taking into account your previous qualifications, the degree committee has decided to grant you the degree of doctor. Professor Roeshop is authorized to confer upon you this academic distinction in accordance with Dutch university custom. And I invite your supervisor to now take the floor.
Do you promise to work in accordance with the principles of scientific integrity at all times, to be careful and honest, transparent, independent, and responsible?
I promise.
By the authority invested in us by law and in conformity with the decision of the committee here present, I hereby confer upon you, Lydie Marie Odette Barbeau, the degree of doctor and grant you all rights attached by custom and law. As evidence of this, I now present you with a degree certificate signed by the rector, the secretary, and the other members of the committee, and affixed with the official seal of the university.
Tu peux venir devant, maman, prendre des photos si tu veux.
Lydie? Lydie, Dr. Barbeau, first of all, congratulations with this great achievement. I know you're really nervous the last couple of weeks, and especially today, but we just all agreed you did a fantastic job. And after receiving your doctorate today, you closed the last chapter of your PhD journey. It's time for a new one. And after almost 10 years, of which almost 5 in our group, you will leave Maastricht for the second time. So you came to the lab almost 10 years ago, where you started in a group of Mark Voice as research technician, where you started working on notch signaling. And I think during that time, you laid the foundation of your practical skills and the mindset that were of great value of your PhD project. And during that time as a technician, we got to know you already as a hardworking and dedicated scientist that you are. But as a technician, you decided you wanted more. You wanted to do your own research, you wanted to make your own plans, so you decided you wanted to pursue a PhD. And for that position you found in Essen, and for the sake of science, you left Maastricht for the first time. But fate had other plans. Meanwhile, a bit more than a year later, we had a PhD position available in our group, and through the grapevine we heard that you were not really happy in Germany. So Casper, cheeky as he is, he offered a position to you, and you were happy to take it, and you could not wait to start the project with us. It should have been a great start. We planned a collaboration with the University of Toronto, where you planned to do your dropout screens, but unfortunately, COVID pandemic started, and so we had to cancel our plans. Huge disappointment for all of us, but of course, especially for you. But we set up a new collaboration with the NKI. You adapted very well, and thanks to your dedication and perseverance, you managed to identify those targets we were looking for. And targets that we will still pursue after you leave. In addition to that, you published some great work. You did a nice review on cancer genotypes that are autophagy dependent. You delivered great work on predicting chloroquine sensitivity in glioblastoma, and identified LCTC as a potential biomarker that maybe in the future can help predicting responsive versus non-responsive tumors. A chapter very close to being published, and plus also in collaboration with Lotte and Nikki. Nikki, sorry. You didn't keep that amazing work for yourself. You shared it at international conferences in Hungary, twice in Norway, And you even won the best poster prize, and even at the prestigious AMBO workshop on autophagy, known for its high scientific level, because your work was received very well, and they even awarded you with another best prize for poster. And I think those are remarkable achievements. In addition to doing science, you're also learning Dutch. And I think that goes pretty well. I am not sure if you are already familiar with the phrase met de Franse slag. And the Franse slag means literally translated with the French strike. It's an old saying, and the Dutch use it when the job is done but sloppy, not really thorough. And so you can imagine historically the Dutch view and the French was not best. But after having you in the lab, I think we have to reconsider that phrase. Of course, you delivered anything but half work. I don't know a lot of people that are as thorough and organized as you are. Your work ethic, 7 days a week, first in, last out, was unmatched. We even had to force you to take a holiday, to take some time for yourself, and even then, you were waiting to go back to the lab again. The sheer amount of data you produced is staggering. We tried to trace back how many Western blots and qPCRs you did, but it was countless. It must have been hundreds. And I think your student Eric, I don't know if he's here, I think he still dreams about those Western blots. And sometimes the universe conspires against you. During renovations, one floor higher, a water pipe broke, and during the night, your desk was flooded, your lab journals were soaked, your computer was fried, but you didn't let that stop you. You took a hairdryer, dried all you dried all your lab journals, and you continued to work as if nothing happened. But of course, like every perfectionist, you find it sometimes difficult to distinguish the essential things, the important things, and the not so important things. And sometimes a bit more often, that caused some stress. And believe me, everyone knew when it was that time that you were in a stressful period, because angry emails were sent to the whole department, guided with pictures and angry phrases of who left the pipette, who didn't clean the hood, and who took the last bottle of medium that you used for your meticulously planned experiments. And I think during those times we also learned all a bit of French, but of course only the best words, which I should not say right now, but since your mother is here, I think she has to know they contained merde, putain, connarde, bordel de merde, and some more. But although your messages could be firm at some times, it came always from the incredible drive to do things impeccably. And in addition to learning some French words, we also learned of some of the best French cuisine has to offer. I still remember your signature dish, poisson de micro-ondes, some fish from the microwave. I can still smell that fish.
That was Venus, not me.
No, that was your fish.
Okay.
And for the best, we haven't smelled it in a very long time. Apparently, you swapped it for chicken now. And I have to say thank you from everyone in the Juns 50 building. Lydie, during the last 10 years, we got to know you as a very kind person, a caring person, a hard worker, and always ready to help your colleagues, even if you didn't have time for it. You always said, yeah, sure, you grabbed your tiny notebook, you wrote down what to do, and you are ready to help. Even if that contained collecting resecting lower jaws containing teeth or tongues with tumors, and isolating tumor cells from that. Thank you for that, again. I never had any doubt about your knowledge, your data, or your skills in the lab. Lydie, this chapter of your life may be closing, but you can look back with immense pride on everything you have achieved and accomplished. We certainly are. The next chapter awaits as you leave academia and Maastricht for the second time to become a study director at Charles River in Groningen. We wish you all the best in this new role, and knowing you, I have no doubt you will do great. I would like to say congratulations once again, and thanks for everything you brought to the lab and to our team.
Thank you.
Thank you, Dr. Curtis, for a very nice laudation. Dear Dr. Barbeau, also on behalf of the Maastricht University, I congratulate you with the degree you have acquired. It's a great day, of course. In French, you would say, cela tient du prodigue. And this is really great. I hope you can celebrate it, and I wish you a good future, scientifically and privately, of course, and keep the balance. That's what we say these days. I also congratulate Professor Ruschop and Dr. Keulis with their with this important promotion, and of course your family and friends here present and online. I would like to thank the members of the opposition for their efforts, and of course the technical support team and the PEDEL, because without them this session would not have been possible. Having said that, I'm about to close this meeting, this ceremony, but we are going to make a picture here with our Groningen professor on the television screen. So I would ask the audience to already go to the rafter where you can have Limburgse fly, I suppose. And then we make here the picture, and then we are going to make a picture at the foot of the stairs. So if you can already go to the reception room, except for, of course, the photographers. And I close this meeting.
Yeah, I will just translate to my family.